And the general ideas is still okay? Do you still hear me?
Yes, sound is coming.
Hello?
Yeah,
Okay.
yes, yes, it's okay.
Okay. Good. So the general idea is introducing fluorescent nucleobase analog site selectively in DNA or RNA sequence in order to sensitively probe local conformational change. And these fluorescence nucleotide analog nucleobase, they ideally should replace natural nucleoside without perturbing the DNA structure and function, keep high brightness, exhibit spectroscopic properties sensitive to the environment, of course, and so reveal quantitative local information on DNA structure and in its interaction with biomolecules. So, as we said, we have to introduce them, and there are two major methods how to do it. So one is a chemical method, so solid state synthesis. So here we have the major step involved, actually the step involved. So how is working in few words. So here you see you have the nucleobase with the bases and the sugar, which has to be where the group initially are, some group initially are protected to reveal their reactivity only when it is required. And the key group is the phosphoramidite, which is this group here. And so these are the major steps. So let's say the start is practically here, and we practically you add a nucleobase one by one. So the first step is so you have the first nucleotide that is linked to a support, and then there will be the protection to reveal the reacting group that are required for continuing the reaction. Then you add, and so then the subsequent, the N plus one, let's N plus one nucleobase in the phosphoramidite form is added, and there is and the coupling occur. And so as you see, so then finally, so in addition, when what did not react is acetylated to prevent further chain extension and so on. So then we have now two nucleotides, and then we start again. So we have the third one and the fourth one and so on. So this is very good, but it's a method that is limited to length of 100 nucleobases. 160 actually, even for RNA. For longer sequence, there exists the enzymatic synthesis of label oligonucleotide. But this method brings, produce lower quantities. Anyway, these are also the steps. So practically we use the machinery of the cell to produce our nucleotide sequence. So here is an example for RNA, but of course DNA is similar. So the central enzyme in this case is the RNA polymerase, which actually, as you see, is as a substrate, so is used. So what is used are the different bases, and in addition also the the base is X, which would be the fluorescence nucleoside analog. And so the polymerase, that is how it works. So is with the nucleophilic attack of a bound nucleoside 5' triphosphate by the 3' hydroxyl of the RNA primer. So then, yeah, that's is. And the result is the incorporation of the nucleoside, monophosphate, and pyrophosphate is released, as is seen here. So at the end, we have increased the sequence of +1 based on a scaffold. Of course, is the complementary, is the scaffold, the template tells us which will be the next residue, the next nucleobase that has to be added, as is expected here. So practically at certain point, and then in case we have a fluorescence analog, fluorescence nucleoside analog, then what we will have is a sort of competition. So let's imagine that here is a analog of guanine. So we will end with, we can have some sequence like this and some sequence with the natural guanine. So purification has to be done to separate the labeled from the unlabeled. And then somehow the rest has to be added when we have the right position. So for doing this, it has to be designed a splint DNA, which actually is again a sort of template that tells us how the donor and where actually the donor and the so that allowed actually the so-called donor sequence, which contain the label. To be connected to the acceptor sequence that contain all the rest of the sequence. And so that is the way to practically to fix the label in a specific wanted position in the sequence according to the splint that is used. And okay, so yes, but of course, I mean, limitation beside the quantity is also the fact that the modified nucleotide has to be tolerated by the polymerase. So then, I mean, of course, some deliberation need to be made. Okay, so so here are the environmentally sensitive fluorescence nucleobase. So are many, many are existing, and actually starting from the 60s, the scientists work on this subject because the need was evident. And as you see, the first, as we will talk about it, is 2-aminopurine, which was, but we will talk about later. So the fluorescence nucleobase analog are divided in 4 major categories. And so we have I start from this side, because we will concentrate more on in the next, we concentrate on the so in green we have the chromophoric nucleoside analog. So here we have practically the entire nucleobase you see here, that is the sugar. So in the place of the nucleobase there is a bulky aromatic chromophore that is fluorescent. And this is, in orange, we have the extended nucleoside analog. So that practically, so there is the fluorophore is tethered to the nucleobase. So, of course, also here we have bulky situation. So extended and chromophoric nucleoside analog exist, and they are used especially when brightness is needed, because they are really particularly bright. And they are useful when we want to detect a particular nucleic acid. But they have a very large size, and so they can compromise the folding and the stability of the level of the nucleotide. They can hinder the interaction with protein. So yes, they are, especially for our purpose, they are not really ideal. Is, for example, expanded nucleobases. So here it is just an additional ring, for example, so the conjugated system for creating a larger conjugated system is added. So then we can produce, let's say, fluorescence, but with a limited dimension. And then the other very interesting, of course, the name isomorphic is, of course, quite appealing, because, and here, sorry, I have to admit there is a problem here. There should be written isomorphic. That's, yes. Oh, sorry. Oops. Okay. Isomorphic, so I insist here on this side. So the idea is practically to transform the structure of the natural nucleobasic in an atom economic way. So really just maybe an anatomy substitute. So and the nucleobase practically maintain the form. And that is actually quite interesting, because normally, so actually this kind of fluorescence nucleoside analog, the base pairing ability is maintained, as we will see. So we will just talk about these two categories in the next, and especially in the isomorphic, on the isomorphic category, on which actually the group in which I'm working is focusing on. And I think that Jagannath also will recognize something about what I will say in the next. So yeah, yes. So that is some example of expanded pyrimidine analogs. So as you see, that's are the natural nucleobase, and these are their expanded version. So, and they have all useful properties, and according to their quantum yield, more or less interesting, or the lifetime, and so on. They can be used, and they found application in this in the study of nucleic acid. So that is the case of cytosine, that is expanded, and here is thymine, that is expanded. So important is to see that in many of them, like this analog or this analog, so you see you maintain the group that are involved in the base pairing. Okay, so that is just here an example I take only one of them. So then is the deoxyloquinazoline-based nucleoside. So it's this one, which water has quite good, actually a good quantum yield and one lifetime. And its replacement in the DNA duplex leads to a limited destabilization. So the melting temperature is just a few degrees higher than the natural nucleoside analog. So it seems okay, and it could be used, for example, so there are differences between that was in water, but in the duplex or in single-stranded DNA, this photophysics is different. It's different according to the stacking, different type of stacking interaction. It's also the photophysics is depending also of the flanking basis. The quantum yield, though, is very low in nucleic acid, and that's actually is a common problem of this fluorescence nucleotide analog. So is anyway higher than the fluorescence, 2 orders of magnitude higher than the fluorescence, intrinsic fluorescence, but is still very low. Anyway, can be used, for example, since it depends from the flanking basis, so you can have also some information about the sequence. And then, for example, this is this particular probe, when it's flanked by ethymine and the A residues, its emission is sensitive to the nature of the opposite base in the duplex. And so this probe can be used, for example, to detect a single nucleoside polymorphism, for example. As you see, so that is the difference when this probe, the opponent is a adenine in green or a guanine in red, for example. And so just between us, so the adenine will be the correct one, and the guanine will be, of course, a sort of mismatch. And so that is detecting this problematic. Moreover, can be used also in from some as a donor in some fret pairing with another nucleoside analog. That is an analog of the cytosine. So good. But okay, we were talking about expanded pyrimidine, and there are also expanded purine analog. And here is an example for adenine so it's an expanded adenine analog. Okay, so let's move to the more interesting isomorphic fluorescence nucleobase analog. And we start with the 2-aminopurine, which was the first to be developed actually as a first analogs that was developed. And is an isomer of adenine, which different from the adenine, you see, from the absence of this amino group in position 6. But it remains still very similar to adenine. And so actually it may form the Watson-Crick base pair with thymine, so that means that the duplex can be formed, but the AT pair is less stable than the canonical one, and it can also isomerize in some wobble pair. So here you see there is a sort of rotation, so it's not the same group that are interested. The two hydrogen bonds are not formed between the same group. Moreover, aminopurine can also H bond to cytosine. No, actually, this is with thymine, but also it can do it also with cytosine, and it forms therefore a mismatch. And the can also form a mismatch with adenine and with the guanine that can distort the local conformation of the bDNA, which is the most common double helix that we know. So here is the relative stability of the base pairing. So they are practically all existing, although less stable, but they all can form, and that is not really a good thing. That is. Moreover, so in general, the incorporation of this pair, 2-aminopurine-thymine pair, preserve the overall double helix conformation, but cause some local changes like narrowing the minor groove. You see that, so in the double helix, you have a major and a minor group, and the minor group is narrowed, even narrowed with the aminopurine. And so, stabilization. Anyway, even if there are some drawbacks, is really the most widely used fluorescence nucleotide analog for studying DNA and RNA dynamics, folding, and interaction with protein, as we can see here in some example. So is 2-aminopurine can be a good, let's say the central principle of 2-aminopurine sensing is the dependence of quenching, of the quenching process on local base stacking. And so the incorporation of 2-aminopurine in DNA or RNA decrease the quantum yield by up to 2 orders of magnitude due to additional relaxation mechanism enabled by the stacking between the bases, and so the electronic interaction between the natural nucleobases. So practically can be detected. So what we can detect in this case, so that is in base stacking. So there is this connection. So the especially charge transfer from guanine, which quench the fluorescence of 2-aminopurine, which instead is restored when some local, let's say, when the sequence is locally, when the base pairing, let's say, that is locally disturbed. And so, as you see, so we can use this to probe local conformational disruption, for example. So it's and anyway, a complex is 2-aminopurine has a very complex photophysics, because the fact that there are all these different quenching mechanisms involved then lead also to multi-exponential decay. And here, for example, is the 2-aminopurine alone has only one lifetime. Instead, in one nucleic acid, here the primer binding site, you see 4 lifetime, plus even a dark species. Of course, with the TCS-PC technique, which we were talking about, so time fluorescence, is very short lifetime is not detected, and that's why all what has a lifetime, let's say, longer than a picosecond, 50 picosecond, is considered a dark species. So practically, this is the contribution of the species that has a lifetime, or more than one, maybe can be also more than one lifetime less than 50 picosecond. So yeah, okay. So then these are the long lifetime is attributed to a extra helical aminopurin, since it's very similar to the lifetime of the nucleoside alone. And the other three, they correspond to different conformation of the nucleotide. With respect to the rest of the nucleic acid structure, sequence and structure. So that's complicate, of course, the interpretation of the data, and can be anyway used also with some, of course, care in the analysis for all the reasons that we said, to study the dynamics and the interaction of a nucleic acid. And in particular, as I said, so it's very good to when we can study this process that lead to the destabilization and the passing from, for example, a complete duplex to a single strand local structure, because there is where where the signal of 2-aminopurin is very sensitive. It's very sensitive and can be more easily interpreted. So, for example, we can study the kinetics of DNA unwinding, in this case, following the binding to the endonuclease Cas9. So that's an example. Okay. So yeah, so then for the same reason also nucleic acid folding and unfolding, we can have some information, and also probing interaction with the nucleic acid binding properties. So as you see, bound and unbound, the fluorescence is different, and so we can follow the interaction. So it seems quite. So in summary, all these examples show that 2-aminopurin can provide, is sensitive to the local conformation of nucleic acid, but in many situations, when it's incorporated in nucleic acid, its quantum will decrease very much. So, which may be a problem for certain type of mechanism that we may be interested in, and moreover as a complex photophysics. So we can maybe do better. We hope so that the scientists try to think about to improve this story. So to find better isomorphic nucleotide analog. And so in the last decade is the thienopyrimidine family was developed. So here is practically the presence of the heteroaroma. So the ring is so as you see. So that is the pyrimidine, which is the basis that we use, and is actually the introduction of this the thienopyrimidine. So it's characterized by the sulfur here, is heteroatoms, and that is actually responsible to the fluorescence signal of this analog. And several have been synthesized, and but only the thienoguanosine and the thienoadenine are really isomorphic. As you see, that are the two that are closer to the natural adenine and guanine. So, yeah, so this fluorescence nucleotide analog show to be very sensitive to the polarity, in particular the adenine and the guanine that we were already underlining to be the better, to have the better isomorphism, as you see. So in two different solvent, water is more polar, of course. The wavelength, there is a change in the emission wavelength, and the factor that indicate the polarity, and actually guanine is better than the thienoguanine, is better than thienoadenine, as you see. So, and moreover, it is also easier to synthesize. And our lab is working with this fluorescence nucleoside analog, and actually made a huge work for characterizing these bases, this compound. So is definitely a true isomorphic analog. So the Watson and Crick base pairing are absolutely maintained. These are the groups that are involved. And if you compare the melting temperature, which is actually a signal, the melting temperature, I don't know if you know, but is a signal, is a signature of stability, because it indicates the temperature at which you unfold practically the sequence. And so let's say higher is this temperature, more stable is your sequence. And normally when you modify the sequence, you decrease the melting temperature And here, as you see, as you can be compared between the black and the orange line, so the melting temperature in function of the opposite base is the same practically in between the natural guanine and the modified guanine. So the photophysics is bit complex, but not as complex as the one of 2-aminopurine. The complexity is due to the fact that there are two tautomers that can form, and they have two different spectroscopic properties. So the so-called H1 tautomer has this kind of profile, that is the absorbance and that is the emission, and the H3 is instead the blue one. they have anyway similar quantum yield. And so this can be this aspect can be also used to probe something, because, for example, if you in duplex, the H1 is favored, and so you see the different signature in of when the emission practically of so the different signature when thienoguanosine is in duplex than when it's in some single strand structure, where there is the contribution also of the other tautomer. So good. So steady state fluorescence so properties are better in thienoguanosine. And so so what I want to say is but is better, you see, so they actually is much more fluorescent than 2-aminopurine, and is actually more sensitive also to polarity change, because the shift in the wavelength is more pronounced. So the concerning the lifetime, as I said, is more simple. So that is just to remind you how was the situation in 2-aminopurine. So for lifetime plus dark species, here in the same situation, so in the primer binding site, the thienoguanosine has 3 lifetime and no dark species. So yeah, okay, so that's is let's say that's we can move on. So here is also in duplex, yes, that is also just the comparison also about what is the situation of 2-aminopurine in duplex and of thienoguanosine in duplex. So also here is the species are much less. So the thienoguanosine adopts a major conformation in the base in duplex, as in agreement with the steady state properties, as we saw that one tautomer was selected, and so of course like a species is taken out from the pool of possibility. So yeah, so then okay. I just, if I may, I just ask you. So before continuing, I need just it's not very long, but just 5 minutes of break. Is it possible? So really,
Yes, of course, of course.
yeah. Okay.
No,
So
no problem.
just a second. I mean, I will remain. I just come. I just I will
Okay.
come immediately.
Fine.
Okay.
Five
So
to 10
really,
minutes.
5 minutes. Not more than this. Maybe even less. Okay.
Okay.
Thank you very much. Sorry. Yeah. That is. I come. Okay. Are you still there? So is
Yes. Yes, sound is okay, clear.
hello?
Yes, are you hearing?
So hello, sorry, I was not, I didn't have audio. So is okay. So everyone is back. S- okay?
Yes, yes, it's
Yes,
okay,
yeah,
and the
okay.
slide is yes, yeah.
Yeah, okay. Sorry, very sorry. I needed just a moment of, of break. But okay, now everything is fine, and let's go on. So I before going on, I just try to say a few words about a very particular aspect of genetics, and which actually is epigenetics. So actually, we may are all surprised the fact that in our cell, in the nucleus, there is the genome. So the genetic information is the same. We have only one genome, and but the type of cell, the type of organs, the type, so are really the tissues. They are completely different, and and so yes, how this variety is possible. And actually, this is epigenetics, which define actually the phenotype without any mutation in the phenotype, without any mutation in the DNA sequence. So actually, the DNA sequence is what it is, but there are so-called epigenetics mark, which are allowed to express or not certain numbers of genes, which determine the identity of a cell. And so practically the epigenetic marks, they tell us how to read the genes. And among the three type of epigenetic marks, there is a DNA methylation. And DNA methylation is a biological process by which a methyl group is added to the DNA molecule, mainly to a cytosine base. As you see, that is the normal one, and that's actually there is an enzyme, DNA methyltransferase, which is catalyzing the addition of this methyl group. And when this methyl group is located in a gene promoter, DNA methylation repress gene transcription, so gives this sign. Normally, DNA methylation occurs in CpG dinucleotide, so when practically a cytosine is close to a guanine. And in mammals, around 70% of CpG dinucleotide are methylated. And okay, so actually that's also is a very delicate point, because in cancer cell, gene promoter, CpG island, so region where practically are, so sorry, I mean, because I was not talking about the fact that there are CpG island that are generally unmethylated and located in gene promoter regions. And okay, so we have some cytosine are methylated, some not. So then, and of course, the methylation patterns is suggesting which kind of gene has to be read. And in cancer cell, the G promoter CpG island acquire abnormal hypermethylation. So even them, practically the methylation is out of control in cancer cell, which is one of the reasons why cancer cell developed abnormally, and actually why cancer is cancer, and that is. So that is an important mechanism that has to be kept, of course, under control, and is also interesting to investigate. And so, just to see how is what is the mechanism behind DNA methylation. There is also these slides, which actually will be a small video. But before, I just want to say that, as we saw, methylation is quite important. And the methylation, the DNA methylation pattern, need, of course, also to be replicated. Because if you want to build a tissue, we need a lot of cell, and all these cells, they have to have the same methylation pattern, because they have to produce the cell that goes all to make this particular tissue, just to say. And so then there are two enzymes that are important. The one that catalyzes the methylation, but also the one that is recognizing, and this is the protein UHRF1. So as we see, so that is what happens when we want to try to reproduce the DNA. Okay, so then there is the annealing, and the little blue mark are methylation. So then UHRF1 arrive, and it's flip the methyl cytosine, which is practically a signal for the dimethyl transferase, so the DNA methyl transferase, which receive practically the message to flip the corresponding cytosine corresponding to the flanking guanine. That's why we talk about the CpG site, which then can be methylated. And so we have copied practically the methylation also in the newborn sequence, and so on. So voila, and at the end there is the pattern which will be respected. So just to say, in the lab, so then as you see, is a dynamic process, and recently we have submitted a paper which is actually under revision, and which I was also participating, and thanking, and we could, following the fluorescence of TAG, we could explain the different steps that occur in the interaction of the binding domain, the SRA domain of the protein UHRF1, which allowed the process of the flipping, which is this process that recruit the DNA methyltransferase to transfer the methylation in the newborn sequence afterwards. And so to study this kinetics, we were using this instrument, which actually is a fluorimeter, because it works like a fluorimeter, fluorescence, excitation, and emission. Okay, but what is has the possibility is called stop flow, this instrument, and you rapidly mix the partner, and you can follow the kinetics of their interaction. So that is the importance, the addition of when I say rapid mixing is in the millisecond range, which is definitely much faster than what we can do if we just pipette in a cuvette and we shake for making the mixing going on. So, okay, so it seems actually that with TAG we can do many things, but can we do even better? And yes, the answer is yes. Because so there were the Thieno pyrimidine family, but with the isotiazolo pyrimidine family, the isomorphism is, let's say, improved, and we can have not only isomorphism then, but also isofunctionality. And the reason is that in this family of fluorescence nucleotide analog, the nitrogen in position 7 is maintained, and that is that is actually the natural basis, as you see. So there is a nitrogen here, which is not there in thienoguanosine, but is there in isotiazologuanosine. So that is the guanosine analog. And that is extremely important because it plays an important role for the formation of DNA, RNA ternary, or RNA tertiary structure, and in the interaction with protein. Okay, so several thiazolo analog were synthesized. Again, the better of all of them. So the real isomorphic are the one of adenine and guanine, but the guanine analog is, let's say, the best because it's the brightest, and he has also is more sensitive to the polarity, and is the probe that we are characterizing at the moment in the lab. So it perfectly replaces guanine, and as again the temperature indicate, but the melting temperature indicate, but is what to say? But in addition, it behaves exactly as guanine with respect to some enzyme like the guanine deaminase, because this enzyme is recognizing the nitrogen in this position. So with the TAG, this enzymatic reaction would not occur. So in this sense, TZG, if we want for being short, is also isofunctional. I realize, sorry, that here are I see it now that they just are the same result that were for TAG, you can imagine that for TCG, actually, they are very similar. With respect to the replacing in duplex are TAG and the TCG are actually both very good. So, which justify the mistake in the figure that I was choosing here. So, sorry. The information is anyway the same, than what I want to say. But as I insist, TCG, in addition, has also this isofunctionality. For example, it can be recognized by certain enzyme. Also here is the enzymatic synthesis of the nucleotide, which is catalyzed by the NCV, that's the cyclic dinucleotide synthetase from Vibrio cholerae. And this again is much better recognized TCG than TAG because of the presence of this nitrogen. And okay, so if you want to compare the fluorescence properties of both, so they are in a certain way similar, with some difference. So both TAG and TCG has the two tautomer, H1 and H3, but with different spectral properties. But you see they are a little bit different, the spectral properties. So then is because in the and in particular in TAG, both tautomer were equally bright, the same quantum yield, and instead in TCG, the H1 tautomer is less bright, yeah, but is the only one that is bright because the H3 is practically dark with a very short lifetime. So, but anyway, TCG, although is less bright than TAG, it shows higher sensitivity to the polarity and the H bond ability of its surrounding, as you can see here in the comparison between the aqueous solution and the methanol, for example, and dioxane. So the quantum yield for TAG is practically not changing, and instead is changing quite a lot in the TCG. So practically, when incorporated in oligonucleotides, TCG is expected to exhibit a more sensitive response than TAG upon protein binding and local conformational change, since it's very sensitive, is much more sensitive to polarity. And so, as you see, the verb is expected is as a reason to be there because it's a very new probe that we are still characterizing, and at the moment we are fighting to find the way to incorporate it in oligonucleotide. In particular, we just recently got successful to synthesize the phosphoramidite, which of TCG, which which, as we know, is important, essential if we want to make solid state synthesis of oligonucleotide. So anyway, TAG is very promising, and not only as also another important advantage TCG with respect to TAG. So, as you know, as you know, duplex, the double helix of DNA, repose on the formation of hydrogen bond between the bases, opponent bases, And this hydrogen bond, which are 2 in the case of adenine and thymine pair, and 3 in case of guanine and cytosine pair, are called Watson-Crick base pairing, practically, are the interaction in Watson-Crick base pairing. But there is also another type of base pairing, which is called Hoogstin base pairing, that involve other groups. So not these groups here, but the other one. So, and this the other phase practically of the, so is this part here of the adenine, for example, and also of the guanine are this this phase, not that one, but this phase, this the group in this side of the guanine that are involved in Hoogstin base pairing, and so is the nitrogen in position 7, which is involved in Hoogstin base pairing. And so it's essential that in TCG, so it's very important that in TCG this nitrogen is maintained. So that means that TCG can form correctly Hoogstin base pairing, and TAG not really, or when it form them, they will be less stable. And where Hoogstin base pairing are important in the stabilization of such tertiary structure of the DNA, and in particular of G quadruplex, which is practically, you see, are formed by a particular folding of the sequence of nucleic acid, and such folding allowed 4 bases to be on the same plane, and the interaction between them stabilize this structure. And they are called the G quadruplex because the 4 bases implied in this quartet are all guanine. And so that means that the G quadruplex can form in sequence rich in guanine, and guanine, of course, they have also to be distributed in a particular order. The group of guanine has to be distributed in a particular order for allow that then you have a guanine in face of another guanine to form the quartet. And so that is an example of the quartet. On a G quadruplex of quartet, or G tetrad, actually, can be also said. And as you see, so here we have inserted a TCG. So these are all natural guanine, and that is a TCG, and due to his nitrogen in position 7, it can form exactly the same hydrogen bond than a natural guanine. So that is a perspective work. If we want to know to study the folding of such quadruplex, which actually is affected, or let's say promoted by ion like sodium and magnesium, sodium and potassium. But TCG will be definitely the ideal form of nucleotide that can be used, and so that will be in the future. Just for your information, we are actually making such study with TAG in this position, and we have some results, and some information can be obtained, but we realize that there are some stability problems that are confirming that THG, the Tieno version, is not the ideal probe for such study. And so we are hoping for the TCG when it will be ready. Okay. So I think actually that I am close to the end, and before I would like to conclude with some remarks that about the wood lecture that I had the chance to give you. And so, yes, I hope that you enjoy, first of all, and I hope also that you could understand that fluorescence spectroscopy is an extremely versatile and sensitive tool. So versatile, there are several types of techniques that are based on the fluorescence signal, and they can be more adapted for investigating one process or the other, and there are and this is a very sensitive, because we can work with relatively low concentration, which are the one that normally we have in biological processes. Sensitive because we can also reveal certain phenomena that without a signal could not be revealed. And of course, is also important to know the basic principle to be able to set the appropriate experimental strategy, and afterwards to interpret the experiment. So that's why I was insisting on certain theoretical aspect. Without knowing them, you cannot go further, or at least you cannot go further correctly. And fluorescence probes are the tools to monitor the conformational changes in biomolecule and their interaction, the interaction between biomolecules. And fluorescence probe, as we saw, there are sometimes intrinsic and sometimes there's to be added, but in an appropriate way, of course. And moreover, it's maybe you understood that the best fluorescence probe is not the brightest. Here I want to say brightest. Sorry, it's not the brightest. It's not the one. Okay, it's a high quantum yield. Okay, I take this because that will be for sure giving me a good signal. No. It is the one that allows to get you insights in the real mechanism of interest. And with this, I actually mean that has to be a probe that is sensitive to the phenomenon that you want to observe. And when you insert it in your system, you don't modify the system. In this sense, it's the real. So that means that a good probe is the probe that provides information without modifying the system. If not, the information is not on the system, the real one, but is on something else. So that is important to keep in mind for make the good choice, and in case to design better probes in the future to open the option of investigation that can be done. And and so practically fluorescence spectroscopy is a sort of is a art as because it imply a lot of thinking, a lot of reflection, and you have also good feeling, have to have good feeling when you interpret and so on. And just to show the artistic aspect, as that's an artistic aspect of fluorescence, is also that you can paint. And so that's is actually I found it quite interesting. So that's a picture that I got from Tien, in lab of Tien. That was the one that was getting the Nobel Prize for the discovery of and the development of fluorescence protein, GFP, starting from GFP. And so then you see, by using the different variant of fluorescence protein, it was possible to drawing such a nice picture. Okay, so thank you for your attention and your patience to to all my long speeches.
Uh, Thank you, Dr. Elisa. It was really wonderful to have you here. I think it was a very, very comprehensive and in depth um, explanation of the concepts and then relating it to the real life phenomenon. I think it was maybe time was too short. You might require another few weeks to have more
Yes,
better way
yes,
to go in it. So, but yeah, it is really wonderful to have you over here on this platform. I think we would have been more fortunate if this course would have been, in offline mode, so in person. So we would have
yeah,
definitely a better interaction. So let us hope for the
yeah.
best. The part two will come as early as possible, and we will have you here in the campus. You will
That's
definitely enjoy the campus side, the area. It is one of the most beautiful part of our country, India. So it
yeah,
is in the laps of Himalayas. I can't compare any two region in this earth, but yes, every region is having its uniqueness, and Himalayas have its own, uniqueness. So you
okay.
would love to come here. So part two,
Thank
I
you.
would love to have you as a offline course instructor for 1 or 2 weeks. So thank you so much. Over to you, Simran.
Okay, thank you very much for you. I stop the presentation, so then we see us. And so yeah, it's yes, yeah, of course, would have been better. In principle, I also prefer when you have human interaction. It's the best. And okay, so this time was more difficult to organize, but yeah, why not in the future? And about the fact that your region is very nice, I heard about. We have also some student that comes from India in our lab and postdoc. So actually, the lab where also Yaganat was some years ago, and they were telling me, "Oh yes, you have absolutely to go there because it's such a beautiful reason." So I will. I will keep this in mind, and I think that I will accept your invitation at certain point.
So nice of you.
Yeah.
So I
Thank
think
you.
we should wait for another 5 6 minutes because dignitaries and other participants will be joining for the closing ceremony. So Dr. Lisa, if you want to take 2 to 3 minutes wait, you can take. I think we would be. I think the participant would should remain online because we would be starting very soon. So Simran, I think you can brief it in a better way.
Okay, ma'am.
Okay, so just yeah, maybe 1 minute. But I still also here actually. Maybe I drink something because, as I said, and yeah.
So the
So
student have any queries, questions to Alicia? regarding the whole course. If you have any questions, queries, and also you can ask that how the foreign university, or regarding any questions regarding the talks you have already listened the 5 talks and the
Okay.
tutorials, 2 tutorials. Okay. So if you have any questions, you have
Yeah.
5 minutes. You can ask to Alicia. Okay. Any queries, any questions? And then we will start our valedictory session of this GAN course.
I will ask the. Should I ask the this thing to join?
So we have a 4 minutes. Yes, yes.
Thank you so much, ma'am, for such a nice talk. So I have an announcement to make that we will be starting with our validatory section very soon. Just waiting for officials and dignitaries to join soon. So please be online, everyone. We'll be starting as soon as possible. Just kindly wait for a few more minutes. Thank you.
Good afternoon,
Good afternoon.
madam. Archana ma'am, good afternoon.
Good afternoon. Good afternoon.
Good afternoon also. Yeah.
It
So
must be good morning, Elisa ma'am. There it
yes,
is. Yeah.
indeed,
So good
indeed.
morning to you.
So
Yeah.
Dr. Lisa, we have with us Dr. Archana Nanoti, who is presently registrar with us. She has an expertise in electrical engineering. So at the backdrop, she's a professor of electrical engineering, but presently she is registrar with us at the institute level.
okay.
And Archana ma'am, she is Dr. Lisa. I think.
Nice to see you, ma'am. Nice to see you. Yeah.
Yeah, nice to meet you also.
Ma'am, I will call FW and director sir, and then we will start, right?
Okay. Okay. No, no problem.
Mm.
Good afternoon, sir.
Good afternoon.
Good afternoon, sir.
Good afternoon.
Dr. Lisa, we have director of the institute, Professor H.M. Suryavanshiji with us, and
Okay.
he is professor of higher academic grade in electrical engineering department of NIT Nagpur, which is a sister institute of the NIT Hamirpur. So, sir,
Yeah.
we have with us Dr.
Yes,
Lisa.
good afternoon.
Yes. Good.
Yeah,
Good
good
afternoon
afternoon.
also.
Good.
Thank you.
Happy to
Very
see you
nice.
here. Nice meeting.
Sir, should we start
Okay.
with?
Yeah, yes.
Simran, over to you.
Okay, thank you, ma'am. So, a very good afternoon to everyone once again, respected dignitaries, esteemed faculty members, speakers, dear participant, and guests. We have amongst us the guest of honor, Dr. Aliza Bambada. So, as we come to the conclusion of this enriching 5-day Gyan course from 13th of May to 17th of May, I would like to take a moment to reflect on the journey we have taken together. So over the past few days, we delved into the intricacies of complex biological systems and the tools and techniques used to decode them. We began with the solid foundation with Dr. Pamita Avasti introducing us to the fundamentals of G quadrupluxes. Dr. Aliza Bombarda guided us through the principles of fluorescence spectroscopy, both steady state and time resolved, with engaging tutorials on practical fluorescence measurements. Also, Dr. Jagannath Kachlyan gave us valuable insights into biophysical techniques and explored the dynamics of DNA, RNA, protein complexes using fluorescent probes. On day 3, Professor Ritu Barthwal took us deep into ligand recognition of DNA quadrupluxes using absorption fluorescence, CD, NMR, molecular modeling, and thermal melting tools. We also explored the general concepts of fluorescent probes for proteins and nucleic acids and their critical applications by Dr. Alisa. The hands-on tutorials and problem-solving sessions further strengthened our understanding, providing real-world context to theoretical knowledge. On this final day, we learned about the application of quadrupluxes in chemistry and nanoscience, followed by the tutorials focusing on the kinetics and dynamics of biomolecules, and learned about fluorescence probes for nucleic acid. So these sessions showcased not only the depth of the subject, but also its broad applicability in modern research. Therefore, I would like to express my heartfelt gratitude to our distinguished speakers, Dr. Alisa, Professor Ritu Barthwal, Dr. Pamita Avasti, and Dr. Jagannath Kachlyan. Your time, expertise, and dedication have truly enriched this course. Also, a big thank you also goes to the organizing team and every participant who contributed to make this virtual gathering a success. Your curiosity and engagement made the experience vibrant and collaborative. So thank you once again. And with that, I hereby request our course coordinator, Dr. Jagannath Kuchlyan, to officially address all the participants and guests in this valedictory session of Gyan course. Over to you, sir.
Thank you, Simran. So it is incredible honor to stand before you today as the valedictory session of the Gyan course, Department of Chemistry, NIT Hamirpur. I am feeling with immense pride and gratitude for these days we have shared together. In the word of Nelson Mandela, education is the powerful weapon which you can use to change the world. As we are standing at the last day of our program, let us carry this wisdom with us. Let us use our education to inspire changes and promote understanding our students to make a better world. Therefore, I would like to thank our Honorable Director Sir, our Registered Madam, Dean, Faculty FW for their valuable support. I would like to thank our guest of honor, Dr. Elisa Bombarda, for her valuable time within the busy schedule. I would like to thank our national expert. I express my profound thanks to our HOD, Department of Chemistry, NIT Hamirpur. I want to express my gratitude to Dr. Pamita Awasthi Madam, local coordinator of this GYAN, for her inspirations. My heartfelt thanks to all the students, participants, research scholars, staff of our department, and the GYAN team for their cooperations. Thank you, everyone. Jai Hind, Jai Bharat.
Thank you so much, sir. Thank you. Next, I would like to welcome the HOD of Chemistry Department, Dr. Kalyan Ghosh, to address and share some views regarding the session.
Am I audible properly?
Yes, sir, you're audible.
Okay. So respected Professor H.M. Suryavanshi, Honorable Director, NIT Hamirpur, and the Chief Guest of this occasion, Dr. Archana Santosh Nanoti ji, Honorable Registrar, NIT Hamirpur, Dr. Elisa Bombarda, University of Strasbourg, France, and the guest of honor of the event, Professor Anup Kumar, Dean FW, and all the respected deans, heads, the faculty members, and the dear participants. At the beginning of this valedictory session of 5-day online Gyan course on design and development of biocompatible analogs to decode the dynamics of complex biological mechanism held during 13 to 17 May 2025. I would like to congratulate the course and the whole organizing team for completion of this course. Special thanks uh, from me and on behalf of the Department of Chemistry to the foreign faculty of this course, Dr. Elisa Bombarda, for her lectures and being with us today. I hope that these insightful lectures delivered in the course will be beneficial for all the participants. Also, the recorded lectures of this Gyan course will be archived so that at any point of time you can go through them as and when required. I would like to take this opportunity and platform to acknowledge my sincere gratitude to our Honorable Director, sir, who is really a source of inspiration to all of us. Sir, because of your ever positive attitude and inspired patronization and encouragement for organizing conferences, STC, FDP, etc., makes a notable and visible positive change in the environment of the Institute than earlier, and many faculty members are coming forward with such kind of activities almost regularly. Also, words are limited for me to express my thankfulness towards our registrar ma'am for all her administrative support. And thank you all for listening me.
Thank you so much, sir. Thank you. Now, next, I would like to welcome our local coordinator, Dr. Pamita Avasthi, to give us an insight about the Gyan scheme. Over to you, ma'am.
Thank you, Simran. So, as a course coordinator as well as a local coordinator, I welcome Honorable Director Professor H.M. Suryavanshi ji, Dr. Archana Santosh Nanoti ji, Registrar NIT Hamirpur, Professor Anup Kumar ji, Dean Faculty Welfare, NIT Hamirpur, and our guest of honor, Dr. Elisa, Head of the Department, Dr. Kalyan Sundar Ghosh, my co-course coordinator, Dr. Jagannathan, and yes, Simran and Ravinder, Aarti, and Ritu. So you all students who were standing like pillars at the backdrop, 24/7, I must say. And dear participants who have joined online, this 1 week Gyan course is basically falls under the aegis of prestigious Global Initiative Academic Network, which is an initiative of Ministry of Education, Government of India, and its aim is to tap into the global talent pool of scientists, academicians, and entrepreneurs by fostering collaboration with Indian Institute of Higher Education. It provides a powerful platform to enrich India's academic ecosystem, advance research capabilities, and bring global perspective into the classroom. So, over the course of a week, particularly this one, around 14 expert lectures and 4 hands-on tutorials were delivered, covering advanced topics at the intersection of chemistry, biology, biomedical technology, biomedical research, etc. The course featured distinguished national and international speakers who shared their expertise and their valuable research with the participants. So I'm very much thankful to Dr. Elisa Bombarda from Laboratory of Bioimaging and Pathology from University of Strasbourg, France, for delivering comprehensive lectures on fluorescent technology and their application, including the design and development of fluorescent probes for studying proteins, nucleic acids, and other things. So, Dr. Bombarda, once again, thank you very much for your time and valuable inputs. And obviously, we will not leave you. So, I'm also thankful to Professor Ritu Barthwal, She shared insight because of 30 to 35 years of experience of her in academics as well as in research. She shared insights on advanced instrumentation techniques, specifically the nuclear magnetic resonance and circular dichroism spectroscopy, and emphasizes the role in understanding the atomic level structure of deoxyribose nucleic acid G quadruplexes, which is I think the latest area these days. All these sessions were recorded and will be submitted to GYAN national coordinators, that is Indian Institute of Technology, Hyderabad. Additionally, the sessions were webcasted live to reach the broader national as well as international academic or research audience. So, as a course coordinator as well as a local coordinator of the GYAN program at NIT, I express my sincere gratitude to National Coordinator IIT Hyderabad for their support extended for organizing successfully all the courses under GYAN scheme. And my special thanks to our Director Sir, Professor Suryavanshi ji, Madam Archana Nanoti ji, Dean FW Office, Professor Anup Kumar ji, for all cooperation and the kind of positive environment provided to us for successful organization of not only this program but all the programs which has been organized under GYAN scheme, whether it is online or offline. And I'm also thankful to all the participants, and I would love to have any other queries. If you have, we would definitely contact to our experts who delivered the lectures here, not only for this course, for other courses also, and would like to connect to them. And thank you so much. And once again, I welcome as well as thank you to all present in virtual mode. So thank you very much. Once again, Jai Hind, Jai Bharat.
Thank you, ma'am. Thank you so much. Now, I would like to welcome our registrar, Dr. Archana Nanoti. Welcome, ma'am. I request ma'am to kindly address and share your thoughts on our course.
Thank you, Simran. Very good afternoon to all of you and good morning to Madam Elisa. Honorable Director, Professor H.M. Suryavanshi ji, who is a constant source of inspiration for all of us. Dean FW, Professor Anup sir. The local GYAN coordinator, Madam Pamita Avasthi. HOD, Kalyan Sundar Ghosh. The course coordinator, again, Madam Pamita, along with Dr. Jagannath Kushlian. The other speakers, the respectable speakers and participants, and Madam Elisa, who is the guest of honor for today's valedictory function. As I always say, end is not the end, it is the new beginning. So I'm sure that all the participants who have attended this GYAN program, which is a wonderful initiative of Ministry of Education, Government of India. I'm thankful to MOE, Government of India, for this wonderful initiative, which brings the global, all the people in the same area of research, and they can share their valuable inputs with each other through the interactive sessions, and which definitely empowers the participants, improves the innovative and research attributes amongst the participants. So I'm sure that all the participants, they must have reached to some new direction, some new innovative approach towards their ongoing research, or some people might have got some new topic of research which they might be confused initially. And Madam Elisa, I would say this is just the beginning, and we look forward to more of such networking, more of such MOUs or some interactive sessions or some kind of student visits over there or like that. I'm sure that the local coordinator and our faculty members will be in touch with you. So I congratulate the organizing team for such a wonderful program in a virtual mode. I look forward to have many more in future, that too in person, not only in online mode, but I wish to see you all in person as well. So congratulations to all of you. Dhanyavad. Jai Hind, Jai Bharat.
Thank you so much, ma'am, for your words. Now, last but not the least, I now invite our guest of honor, Dr. Aliza Bombada, to kindly share her experience.
Simran, we have, I think, Dean FW with us.
Okay, ma'am. Okay, so now I would like to welcome our Dean, Professor Anup Kumar. I request Sir to kindly share your viewpoints regarding the course. Over to you, sir.
Thank you, Simran. Honorable Director, Sir, Professor H.M. Suryavanshi ji, Registrar Dr. Archana Santosh Nanoti ji, very key resource person for the present valedictory session, course coordinator as well as local GAN coordinator Dr. Pamita Avasthi, head of the department Dr. Kalyan Sundar Ghosh, other young course coordinator Dr. Jagannath, and other participants. Dr. Pamita has given or explained in great details what has transpired in last 1 week. So a lot of areas have been touched, and definitely this initiative of the Government of India is very praiseworthy. And because knowledge has no boundary and it has to be shared across the boundaries so that the global community benefits from that. So definitely that objective must have been achieved, where the expertise of the resource persons as well as those who were attending this course, they must have deliberated on topics of mutual interests. So many such courses have been organized in this academic session. I believe that perhaps for this academic session, this one was the last. I congratulate the local GAN coordinator for facilitating these events, as well as this one, of course, he was organizing as well. So I wish that such courses does happen in future also, and the learning is enhanced everywhere. Thanks once again for giving me this opportunity to share my views. Jai Hind, Jai Bharat.
Thank you so much, sir, for your words. Thank you once again. Now, I request our guest of honor, Dr. Alisa Bombada, to kindly share her experience and share her views for the course.
Okay, so first of all, I would like to thank all of you for the invitation, and then that was a pleasure for me to participate. Yes, so it was quite actually dense, let's say. We can also say demanding, because of course to prepare so many hours of lectures was quite some work. That's I think that all of you that you are preparing lecture, you know what does it mean. And so, but was actually very much. So it was actually giving me something also in this. So that's just while I was assembling the argument, I was also, let's say, myself getting even more interested, even more enthusiastic in the sense that, and so it was allowed me also to watch, to see. To see again some work which maybe was some years ago, and so on, something like this. You forget. No, you make a synthesis, and so that was really. I have to say also that was also formative for me. So then, and this is actually, I think it's the beautiful of teaching, is that this. Between teaching and learning, I would say that the separation is really extremely narrow. I would almost say doesn't exist. So it's really just the two faces of the same thing, and which actually what is the things is to promote knowledge, and that's as a teacher. As a student before and as a teacher now, I am very glad that I can contribute this very important aspect, which is knowledge, which I think that promoting knowledge and knowledge in our world, it will be fundamental. And I also, yeah, that's just I extrapolate, and I say that there will be a better life if there will be a better knowledge, and that's that's we can say. And okay, so, and practically, I have to say, so maybe for me there was some initial difficulties of knowing really the context in which my lecture belongs to. I think that now is a little bit more clear, but and so that somehow I was not sure how deep I should go, what if I should insist on certain things or certain other things, and that was somehow a sort of difficulties that I have, which I was trying to correct on the way of this week, and I hope that that my style of presenting was anyway appreciated and so on. And yeah, then I excuse myself that actually I maybe was the reason why this course was done as a video, online. And I am the first one that believe that human contact are more important and more richer, and so I thank you to have accepted this because it was not really possible for me to organize this way. But considering this aspect, I will take care. I will consider the option to come in person and also to meet all of you in the future. And so. Moreover, I just say to Dr. Arhana, that's because I see that you were talking. It was you that you were talking about G quadruplex. Was your and so that is something that actually yes. There is also a
yes.
Who was Dr. Pamita?
Yeah.
Dr. Pamita. I'm sorry. Okay. So yeah. Yes. And so yeah, as you saw, there is there can be some overlapping and some common interest, and that is also the nice things of this organized course where they keep brings together a person that have similar interest, and so then new cooperation maybe can be also settled down. So thank you very much for this opportunity.
So thank you, Dr. Lisa. We would be sending you a very comprehensive report that what has been done and how we can connect and reconnect and how we can have you in offline mode over here. So
okay.
Yes, over to you, Simran.
Okay, thank you so much, ma'am, for such nice words. Your words really marked great impact on all of us. Thank you so much. Also, I would like to welcome anyone from participant side. If someone is interested to share their views or opinions or thoughts on the course from the participants, if anyone is interested.
Nitesh is here. Nitesh Kumar, yes, please.
Thank you, ma'am. It was really very new experience for me. I have attended almost all the sessions, and it was although different from my research area, but I have listened all the sessions, and all the sessions were very informative, knowledgeful, and I have tried to understand the different biophysical techniques which various resource persons have described in detail, like fluorescence, UV, CD, and really it was very wonderful experience for me. I would like to thank all the resource persons and organizers for giving me this opportunity to attend this course. Thank you all the organizers and resource persons.
Thank you, Nitesh. Any other participants?
Okay, thank you so much for sharing your words. Also, I have one important information that we will be sending feedback forms to all the participants. So kindly give your responses as soon as possible for issuing your respective certificates. Thank you. And now, as we close our web course, I hope this course has sparked new ideas, opened doors for future collaborations, and strengthened your understanding of the biological mechanisms that we strive to decode. So, thank you once again, everyone. Thank you, all the respective honorable guests. So, with this, I formally conclude the Gyan course. So,
Thank you.
Thank you. Thank you, everybody. Thank you.
Thank you.
Thank you.
Thank you. Thank you.
have a great day and stay inspired. Thank you.
Yes. Yes. Bye. Bye. Stay
Thank you, sir.
safe and have a nice day. And
Thank you. Thank you, sir.
Okay, thank you.